Hello. So you can see my screen, right? Okay. So I'm going to talk about the phytoplankton sampling. The samples were processed by Ed Flips and Susan Badalak at University of Florida. And so I'm presenting this on their behalf. So samples were collected weekly in nurseries. So, you know, once your animals are in the nursery, you're feeding them with whatever is coming in with your water. So samples were collected weekly and preserved, archived. Because this process is so labor intensive, we only processed 1 or 2 samples per month, but the other ones were archived, so we could go back if there was something we wanted to look at. And up to 10 samples per facility per year were processed. So those were shipped to the University of Florida lab. The samples are settled in some little special chambers, and then painstakingly, the phytoplankton cells are identified and counted by someone who has years of expertise in identifying those species. For each species, the volume can be calculated or estimated based on, you know, what species it is, what we know about that. And then also we can estimate the carbon amount that is in each of those species. So phytoplankton can kind of be lumped into 4 groups. Dinoflagellates are one of the big groups. Many species of dinoflagellates are good sources of food for shellfish. They store starches and they have lipids. Others, however, do produce toxins, and as you're familiar with, they are responsible for red tides, but that's only a few species. These phytoplankton vary from about 2 microns to more than 200 microns. So if you took a pencil and made a little quick dot on a piece of paper, that's around probably 200, 250 microns, to give you an idea of what that size is. Diatoms is another big group. So many of these species are also very good food for shellfish. They have high levels of lipids or fats in them. Some, however, do have long spines, like this one here, or they form long colonies attaching together. And so those might be difficult for shellfish to filter. Cyanobacteria or blue-green, and diatoms are 5 to 200 microns. Cyanobacteria or blue-green algae, many of these species are also good sources of food for shellfish. They have a high protein content. Some freshwater species, though, do produce toxins, but we would not expect to see those in the marine environment. They are often very small, though. Some of them are smaller than 1 micron. They also can form colonies. So some of these might be actually too small for shellfish to filter out. And then there's another group, which is kind of a bunch of other things get lumped in. Many of these are green things: chlorophytes, cryptophytes, chrysophytes, euglenophytes. They're usually pretty small. They can be good sources of food for shellfish, having a high lipid content. Some species have been known to form blooms and have been linked to fish mortalities in other locations. So I'm not going to show you all of the data, but I'm going to show you some examples. So phytoplankton varies both monthly and annually. Let me see if I can get this thing out of the way. I don't know if you guys can see that, but I can. Okay, so For example, this is a particular site at year 1 and year 2, and you can see that these years are quite different. So in the first year, for example, in the early spring, there were dinoflagellates, and then later spring, we had diatoms. Then in year 2 at that same site, we had a lot of diatoms. In the summer, we had a spike of some other stuff. In this case, it was, let's see, nano-eukaryotes. And so clearly phytoplankton varies monthly and annually, just depending upon what is out there in the environment. Now, phytoplankton also varied by facility because they're in different locations, they're drawing from different water. And so here we have site W329, we have site C578. This one in year 2, for example, had a lot of diatoms, and that varied depending upon the year and the site and the month. So let's pick apart one of these graphs. So if we look at this graph, they're labeled with some letters. And so here we start out in the fall of 2020 with quite a few cyanobacteria, and those were picocyanobacteria, which are very, very tiny. So they may be too small for shellfish to filter out. Then later in the year, we see that we have diatoms. So we start out with C. pelagica and D. fragilissimos. Later, we get some spikes of skeletonema, and then finally some leptocylindrus. And all of these are acceptable food items for shellfish. Now, we also have, when we had the skeletonema spike, we also had some euglena, which is a fairly large other in the other category, and probably can be filtered out. And then later in the spring, we had some nano-eukaryotes. Those are probably these really tiny ones, and some of those might be too small for shellfish to eat. So another way that Dr. Flips looks at this data is that he takes all of the raw data for a particular facility over the entire 2-year study period, and looks at the biomass of every species on every date that he examined. And he ranks the biomass 1 to 50. So he gets this top 50. And then you can ask, well, how many times does a species appear in that top 50 over those 2 years? And that gives you an idea of how important that species might be at that location. And you can also ask, well, are any of those potential harmful algal blooms species? So here's an example of the top 50 graph. So these picocyanobacteria, for example, occurred in the top 50 9 times over the study period. And those might be a little small for bivalves. And we also see that there were some potential harmful algal bloom species occurred in the top 5 biomass 5 times over the study period. So this one, Peridinium, has been associated with harmful algal blooms in other locations, and Prorocentrum has been associated with shellfish mortalities in Chesapeake Bay. So there is a website, What Do Clams Eat?, that was a project from quite some time ago that has tons of information about all these different species and so on. So if you wanted to, you know, pick apart one of these other graphs and learn more about each of these species of algae, that website likely has some information about it, whether it's acceptable item, whether it's not, and so on. And I can try to answer some questions, but the best folks to answer questions would be Dr. Flips or Susan Badalak.
Question, what cell size is at the high end of what a clam can eat?
That's a pretty good question. So it's, I would say probably around probably around 200 microns, 150, 200 microns.
Thank you. No other questions.
Okay,